Hello everyone, welcome back to my channel, igsce Biology Help. In today's video, I'm going to be solving the Biology February-March 2023 Paper 6 Alternative to Practical. Make sure to subscribe and like my videos in order to get more videos like this one. Let's start solving. Question 1. Potato cells take up methylene blue dye when the potato tissue is placed into the dye solution. If the dyed potato cells are placed in the water, the dye will diffuse into the water. A student investigated the effect of temperature on the diffusion of methylene blue dye from the dyed potato cells. So as I have explained in my previous paper 6 videos, anything that comes after the word the effect of, this indicates that the thing that follows this is actually the independent variable. The students use this method. Step 1. You are provided with a beaker of cold water, a beaker of warm water, and a beaker of hot water. Step 2. Measure the temperature of the water in the beaker of hot water. Step 3. Label one test tube C, one test tube W, and one test tube H. Step 4. Draw a line 9 cm from the base of each test tube as shown in figure 1.1. So, over here, this is a controlled variable. This is because this is done to every step. So this is a controlled variable. Place the 3 test tubes in a test tube rack. Step 5. Fill the test tube C with the cold water up to the line drawn in step 4. Put test tube C into the beaker of cold water. Step 6. Fill test tube W with warm water up to the line drawn in step 4. Put test tube W into the beaker of warm water. Step 7. Fill test tube H with hot water up to the line drawn in step 4. Put test tube H into the beaker of hot water. Step 8. You are provided with 3 potato cylinders. They have been soaked in methylene blue solution and then rinsed. Place the 3 potato cylinders on white tile and cut the 3 potato cylinders to approximately 2 cm in length. So, over here there's a source of error. This is because you can't cut approximately, you have to cut exactly 2 cm using a ruler. So this is a source of error if they ask us in the following questions. And also this is something that is a safety hazard over here. Step 9. Place one of the potato cylinders from step 8 into each test tube. Step 10. Start the stop clock. Leave the test tubes for 15 minutes. Again, this is a controlled variable because this is something constant and it is done to all test tubes. Step 11. After 15 minutes, measure the temperature of the water in the beaker of hot water again. Figure 1.2 shows part of the thermometer during step 2 and step 11. Record the temperatures on the thermometer for step 2 and step 11 in figure 1.2. Many students made a mistake in this step 2 answer. They wrote it down as 62, but this is actually an incorrect answer because it's supposed to be 61, not 62, because two of these dashes, they make up 1 degree. So our answer is supposed to be 61. This is 62, this is 63, and so on. Please make sure to focus on these small things. Let's see step 11 over here. It is going to be, this is 36, this is 37, this is 38. So our answer is going to be 61 and 38. Step 12. Remove the test tubes C, W, and H from the beakers and shake all the test tubes for 10 seconds. Now, this is an error to shake all the test tubes. You should not shake it because there can be spilling and then the experiment will not be accurate. Observe the intensity of the blue color of the liquid in test tubes C, W, and H. Again, step 13 has an error because you're not supposed to observe the intensity of the color using your eye. This is something subjective. This means that this is something that can be different from one eye to another. You should use a colorimeter instead. This is a very common mistake or error that is in paper 6 exams. You should use this device called colorimeter to actually observe any color. Figure 1.3 shows the student's notebook with their observations. Prepare a table and record the results shown in figure 1.3. So over here, this is a table that will get you the full mark. This is because it has the temperature of all the test tubes and it also shows the color intensity. So this is a way to draw it. You can use any other valid way, but make sure to use a pencil to draw the tables And also make sure to use a ruler when drawing the table. This is something that I have discussed in my paper 6 videos. State a conclusion for these results. As we can see that as the temperature increases, the rate of diffusion increases, and this is proved by, over here we can see in the cold test tube there was a lower color intensity, and in the warm the color intensity increased. Over here it became from pale to medium, and in hot water the color intensity was very high. So this is something that can explain the high diffusion rate. Identify one possible source of error in step 8. We can say that rinsing may remove the dye. Over here in step 8 they have said that there was rinsing, and the rinsing can actually cause some dye to bleed out, so this can cause an inaccurate result. Our second point is that the cutting is not of equal length. Over here we can see that in step 8 when I was reading, I marked down the word approximately 2 centimeters in length as something that is a source of error. This is because they should be exactly 2 centimeters, not approximately. V, identify one safety hazard in the investigation. Of course, they were using a scalpel. This is something that is very sharp and it can cause some injuries and it can cause cuts. Of course, using cutting board is another safety hazard. VI, the readings on the thermometer in figure 1.2 show that the maintenance of the temperature of the water during the investigation was a source of error. Suggest an improvement to reduce this error. Of course, this is the thermostatically controlled water bath. This is something that I've again explained in my paper 6 video. I really recommend everyone to watch it if you want to really understand paper 6 basics. The student did not repeat the investigation and only collected one set of results. Explain why it is better to collect several sets of results. Of course, this is to identify anomalies. This means that if there is a number that should not be in this result or the range of results, then it can be identified so that the result can be accurate. To calculate the average, of course, we repeat to calculate the average using the mean. B, a student investigated the effect of surface area on diffusion. The surface area is the independent variable and the diffusion is the dependent variable. The student used this method. Cut 4 cubes from a potato. Each cube should be different size. Of course, this is an independent variable again. Put the potato cubes into a methylene blue solution for 24 hours. After 24 hours, remove the potato cubes from the solution and rinse them in cold water. Fill 4 test tubes with water. Place 1 potato cube into each of the test tubes. Leave test tubes for 15 minutes. This is a controlled variable because you're doing the same thing for all the test tubes. Some of the methylene blue dye will diffuse out of the potato cube into the water during the 15 minutes. Shine a light through the water into the test tube after 15 minutes. Measure the percentage of light that is absorbed by the methylene blue dye in the water in each test tube. This is a very important point. It says that the higher the concentration of methylene blue dye in the water, the greater the percentage of light absorbed. So, I say state the independent variable and the dependent variable in the investigation. This is something that we have marked down when we were reading. It is the surface area and the percentage of absorbed light. State one variable that was kept constant in the investigation described in 1b. This is of course the time. You can also say the amount of time that the potato cubes were soaked in, or you can just say time. You can also say the same methylene dye concentration. Figure 1.4 is a graph showing their students' results. Using figure 1.4, estimate the percentage of light absorbed by the methylene blue dye when the surface area of the potato cube is 18 centimeters squared. Show on figure 1.4 how you obtained your estimate. Now over here, this answer can vary. There's a range of answers that can work. For example, it turned out 68 for me. Here's how I got it as 68. To draw something like a tangent, I'm going to go from the surface area of the potato that they said in the question was 18 centimeter cubed. I'm going to draw a line from the 18 centimeter cubed up until the graph, and then I'm going to find out the value in which the line that I have drawn bumped with the line of the graph. I'm going to draw another straight line with my ruler. Of course, you have to use a pencil for this type of question. And I got the value 68. Some people may get it 67. Some people may get it another value. This is something that depends on the printing of the paper. My answer was 68. 2. A scientist investigated the nutritional content of fruit juice. State the name of the reagent or solution that would be used to test the fruit juice for starch. Of course, this is the iodine solution. And double I says state the name of the reagent or solution that would be used to test if the fruit juice for reducing sugars. This is the Benedict reagent. Triple I. A sample of fruit juice was tested for protein. State the result of the positive test. And of course, we use the Biuret solution. It will turn from blue to purple. B. Fruit juice contains vitamin C. Plan an investigation to determine the effect of temperature on vitamin C concentration in fruit juice. Now, as I have explained before, to write any experiment in the paper 6, you must first write the independent variable and then the dependent variable and the methodology, which means the method you're going to use, and also the control experiment over here, and you're going to write down the repeats, which means you're going to write repeat and take average, etc. So first of all, the independent variable for this experiment was using different temperatures, because over here they said that the effect of temperature, which means this is an independent variable. Dependent variable was the volume of DCPIP added to constant volume of fruit juice, or the opposite, which means that I can say that the volume of fruit juice added to constant volume of DCPIP. So you can use either this or that. Methodology: 1. Maintain temperature using thermostatically controlled water bath. You must write down the whole sentence of thermostatically controlled water bath. Never ever just write water bath, because water bath is just adding water in a beaker and then you're going to add the test tube without maintaining the temperature. So you must write down thermostatically controlled water bath to get your mark. Number 2, add 3 different test tubes in different temperatures, 20 degrees, 40 degrees, and 60 degrees, or any range of temperature that you want. And a very important point for this point, please make sure to add equal ranges of temperature, which means that if, for example, here in between 20 and 40 degrees is 20 degrees, and between 40 and 60 is 20, so you must leave an equal gap between the temperatures so that it can be as accurate as possible. And you're going to add this 3 different test tubes in different temperatures with constant fruit juice concentration, or DCPIP. Measure the volume of DCPIP taken to change the color from blue to colorless. This is titration method. You can write down titration if you have learned it in chemistry. It's the same thing over here. So this is basically titration. You're going to see how much DCPIP it took to change color from blue to colorless. And the more volume, the more vitamin C content that is available in this fruit juice. Control experiment. You're going to repeat the experiment using fruit juice, but without vitamin C. So you can see really how much DCPIP will it take to change color. It should not change color because there's no vitamin C, of course. Put results in a table and plot graph, or you can say repeat and take average. C. Scientists wanted to find out if drinking beetroot juice lowers blood pressure. So drinking beetroot juice is actually independent variable. Lowering blood pressure is dependent variable. 2 groups of men had their systolic blood pressure measured. Group 1 drank 500 centimeter cubed of beetroot juice, and Group 2 drank 500 centimeter cubed of apple juice. After 6 hours, their systolic blood pressures were measured again. So after 6 hours is a controlled variable. And this is the data that they were showing. Using the data and table 2.1, calculate the percentage change in the mean systolic blood pressure for Group 1. So over here we can see that before drinking, their blood pressure was 132.4. After 6 hours, when they drank the beetroot juice, it became 127.4. To calculate the percentage change, I have used this method where you're going to subtract the new number from the old number, and this number that you're gonna get, you're going to divide it over the old number, which was before drinking, and multiply by 100 to get the percentage change. This will give you negative 3.778. You're going to round it to one decimal place because they asked us in the question to round it to one decimal place. You're going to get negative 3.8%, or you can just write down the 3.8% without the negative. State one factor that was kept constant in the investigation described in 2C. This is actually the volume of juice. D. Figure 2.1 is a photomicrograph of a section through a bronchiole in a human lung. Line PQ represents the width of the bronchiole. Measure the length of line BQ on figure 2.1. So over here you're going to use a ruler on a paper, and you're going to measure this line, and you're going to see how much centimeters it is, and you're going to convert from centimeters into millimeters, because over here they are asking about millimeters, not centimeters. So over here I got 9.6 centimeters. You can get another answer, so this depends on your printing of the paper, and I got 96 millimeters. Calculate the actual width of the bronchiole using the formula and your measurement. Give your answer to two significant figures. Space for working over here. So I have explained the IM triangle in a lot of videos before. This IM triangle helps you to calculate anything that has to do with magnification. So they're asking about the actual width, so we're going to be calculating the A over here. I'm going to be using the image size. The image size is actually 96 millimeters as we have measured using our ruler up here, and magnification is actually written down here. It is times 130. So you're going to divide 96 over 130. You're going to get 0.74 millimeters. Make a large drawing of the layers of tissue in the bronchiole shown in figure 2.1. I have drawn it this way. You have to draw over here. There's a lot of details. You have to make sure you follow over here. You're going to count down the number of these things over here, and you should draw those swirly things. You should draw them like this, both of them stuck together in this area. You have to make sure you follow the details as much as you can. You must draw only the outlines. You don't draw any cells. Scientists investigated the effect of exercise on breathing rate and heart rate while running. The effect of exercise is actually an independent variable. They measured the percentage increase in breathing rate and heart rate from the resting rates during a 4-minute run. The results are shown in table 2.2, and using all the data from table 2.2, plot a line graph on the grid of the percentage increase from the resting rate against time. You will need to plot breathing rate and heart rate as two separate lines on your graph. So you're going to also include a key. So this is the graph that I have plotted. Over here I have labeled the x-axis and the y-axis. This is a very important thing to do over here, and I have left equal gaps between the numbers and also equal gaps between the squares. So over here I have used 10 squares between each number. You have to make sure to follow this. If you're going to leave equal gaps between each number, and the numbers themselves, they must be, for example, 60 and 80, and for 80 and 100 and 100 to 120 and so on. So they must be equal in gap, and the numbers should have equal gaps as well. And also the same applies for the x-axis down here. You must label it, and you must also add equal gaps. You must start from the 0 point over here. You must start from the 0. And the key, they have asked in the question for a key because you're going to draw two separate lines. You must make them different from each other. So over here I have plotted the points. The points are from the table over here. You're going to, the time of course is constant on the x-axis. The y-axis changes. You can use it as you're going to do a graph for the breathing rate and another one for the heart rate. This is what I have done. I have plotted down the points first, and then I have done a neat line with a ruler, and you're going to use a pencil for all of this graph, of course. So make sure to follow all the rules and also include the key. You're going to, for example, if you want to do another key other than what I have done, you can draw a straight line like this, and then you can add like a dashed line like this, a dashed line like this. And this is another key you can use. If you, for example, if they ask for a third key, you can use the dashed line. So this is all I have done for the table. The last question, double I, state a conclusion for the data shown in your graph. As we can see here that the heart rate was actually increasing with a constant rate, and the breathing rate does not increase with a constant rate. There is a different rate, but the heart rate increases steadily. Over here it is a straight line, as we can see over here, it's a straight line. And the breathing rate is not a straight line. You can also say that as the exercise duration increases, the percentage increase in breathing and heart rate increases. This is another point you can write down. This was the end of our exam. Make sure to subscribe to my channel and like my videos for more videos like this one. Thanks for watching.